Hello, everyone. This is my presentation on lithium, the role of lithium in bipolar disorder treatment. I'm very excited to share what I've learned, and I hope you enjoy my presentation. So, I really want to quickly run down through the different types of bipolar disorder. Bipolar 1 disorder has a manic and a depressive episode with mania characteristic of talking excessively, these euphoric moments, less sleep, delusions, and the depression is the complete opposite with extreme fatigue, prolonged sadness, and bipolar 1 disorder is what most people think of when they think of bipolar disorder in general. Bipolar 2 disorder has hypomanic episodes instead of mania, just regular manic episodes, and they're just less intense than the regular manic episodes. And then I put cyclothymia on here, but in reality, it's not as relevant for my research. But I will say that it does. They just do not meet the DSM-5 definitions for their respective definitions of hypomanic and depressive episodes. So I will be talking about the background and significance of bipolar 1 disorder and the pathological mechanisms. Um, so Similar to schizophrenia, due to the lack of genetic information, it's difficult to accurately study bipolar disorder. Right? It's complex disease with diagnostic heterogeneity, which I talked about briefly before with schizophrenia, in that it's very difficult to deduce genetic abnormalities with just one gene. Right? And it's not one gene that's responsible for all of bipolar 1 disorder pathological mechanisms. And so Given that there were some clinical studies that showed monoamine signaling and disruptions to the hypothalamic pituitary adrenal axis with pathophysiology of bipolar disorder, which makes sense because hypothalamus these are structures that you'd expect the function to be disrupted in a bipolar 1 disorder pathology. And then at the very last bullet point, I just put a couple brain areas that were areas of interest for bipolar 1 disorder. This is a graphic I really wanted to quickly go over because I think it's a very cool graphic that shows what role lithium plays in the mechanism. And as you can see, it mostly affects the cellular cascades or cellular mechanisms, and that, of course, will affect everything either above and below it. So it is just a nice way to visualize this. And don't forget that lithium's ability is to stabilize the mood or mood regulation, and so and that makes sense at the cellular level that it would stabilize the mood through those cellular reactions and how it then affects the systemic and the behavioristic model in humans. So my central question is, how can the treatment of lithium affect the pathology of bipolar disorder and treat the underlying cellular mechanisms for efficient medication? So my first aim is to figure out, okay, given that I know that lithium is a mood stabilizer, what biological molecule is responsible, or is at least strongly responsible, for a lot of the issues that you see in bipolar disorder. And I want to measure GSK3 levels after behavioral testing. So my experimental methodology, according to a study by Rodrigo Vieira, GSK3 it plays a giant role in mood stabilization because of apoptosis, gene transcription, synaptic plasticity. It's just a very key player in bipolar 1 disorder. So given this information, I want to do a social interaction behavioral test to track any abnormalities within aggression or less aggression. Similar to what we did, Dr. Kisner, with what we did with ELS, I want to do the same thing, but with a knockout gene for GSK3 in a control group. And I think a swim test will also strongly visualize the difference between the two, the control group and the GSK3, given its ability to understand whether the mice is going to continue swimming due to elevated dopamine levels or decreased dopamine levels, which will show less perseverance for the mice to continue swimming. So I just put a little graphic here to reinforce what I said. Right, climbing, struggling, inert for the swim test, which would be important to kind of behaviorally figure out the control group versus the knockout, and then the same thing with the interaction with the social performance. And so here I just really want to quickly go over the data. This isn't the data that from any of the experiments that I would be conducting, but this is the kind of format that I would like and sort of how I would portray my information, right? I would show GSK3 activity with the wild type and then the knockout, but I would also show the Western blot analysis of the wild type and the knockout to show for the target protein GSK and essentially what it would look like. I also would decapitate and instantly freeze the tissue after the decapitation and do immunohistochemistry labeling with antibodies on the target protein. And for the swim test, it's very straightforward and just behavioral observation. So my second aim is to determine the neuroprotective effects of lithium with two factors: pathological apoptosis and excitotoxicity, both of which I will define in the next slide. Excitotoxicity is high levels of glutamate or high levels of any excitatory amino acid that will essentially create an issue for the reactions with excitatory neurons. And apoptosis is cell program death, or uncontrollable apoptosis would be absolutely an issue. And so lithium shows great promise because it's very highly sensitive, which it allows it to do its job correctly and allows it to find out the right reactions. And its low specificity allows it to also have restricted side effects on its biological effects for targeting neurotrophic pathways. And so in my research, I would try and find, I found within a paper about how NMDA receptor mediated calcium influx, lithium can protect against that by inhibiting these NMDA receptors. And so it would be interesting to run in vitro models of immunohistochemistry to stain and label those NMDA receptor proteins, while also measuring the calcium and the presence of glutamate in order to find out and make sure that we're measuring the excitotoxicity by quantifying, what will cause cell death, right? The Western blot would accompany the labeling in order for proper identification of the protein. And as shown in my data, or in the kind of data I would like to present, I would like to show a Western blot analysis regardless. And then with apoptosis, I found out that I didn't find out, but I found it through research of a paper that neuroprotective effects in apoptosis for cerebellar granule neurons, they block dephosphorylation of both protein kinase and GSK, and that shows those neuroprotective effects. And this would fit well with my previous study about GSK3. So my expected outcomes, as we went over for my AIM-1, it was a lithium effect on GSK3, and we found out that GSK3 is a major proponent in the role for bipolar disorder pathology, and I would like to see a significant correlation between mood stabilization and lithium treatment mechanism, especially with that of with GSK3. And so AIM-2 for the neuroprotective effects, it was interesting seeing some of the apoptosis, for example, apoptosis preventing dephosphorylation of GSK3, and if we kind of do that experimental method of blocking out the NMDA receptors, I'd love to see maybe that the excitotoxicity could be mediated and therefore can show potential future uses in lithium treatment, not only for bipolar disorder, but for excitotoxicity that may be found in other neuro diseases. And something I'd like to see in the future is maybe like if this is the role lithium plays in BD or bipolar disorder, I would love to see what other role it plays in neuropsychological diseases given its effect of, neuroprotective effects and its effect on GSK3. Here are my citations. Thank you guys so much for watching, and let me know if you have any questions. Thank you guys.